ఇప్పుడు అన్మ్యూట్ అయింది కదా.
Yeah, good morning.
Good morning, Pratita. Hello?
Yeah, ma'am, I'm there.
Yeah. Already participants are joining. And Saber join అయిపోయినట్టు already.
Good morning to everyone.
Two devices నుంచి ఎవరో ఓపెన్ చేసిండ్రమ్మా?
Yeah, it's me, ma'am. Sorry, ma'am. Good morning to everyone. I welcome all you to the webinar. We'll start the session in 5 minutes. I request HOD ma'am to start the session.
Respected dignitaries, Dr. Saber Hussain, Professor of Pharmacology and Toxicology, Wright State University, Dayton, PSA. Professor Sudarshan Reddy, sir, Secretary cum Correspondent, RBVR Women's College. Dr. J. Achyuta Devi, Principal, RBVR Women's College, dear faculty, and my dear students. Very good morning and a special good evening to today's resource person, Dr. Saber Hussain. I, Dr. Chandra Pradyuta, heading the Department of Zoology and Microbiology, welcomes you all for one-day webinar on career opportunities in life sciences. Someone rightly said, we can't expect young people to dream if they don't know what they can dream about. This quote is very relevant to today's program. This webinar aims to provide insights and information on various career paths available for students in life sciences. Professor Saber sir had a very humble beginning and now he is one of the top scientists in the field of biology. We are very pleased to have you here, sir. I'm very sure our students will be able to draw inspiration from your words today. And before further ado, I request our principal, Dr. J. Achyuta Devi, to give welcoming note to this virtual gathering.
Thank you, Pradyuta. Good morning, everyone. I welcome all of you. I welcome students, staff of other colleges, and RBVRR Women's College. I extend special welcome to Dr. Saber Hussain, Professor Pharmacology and Toxicology, Wright State University, USA. I thank Dr. Saber Hussain sir for accepting our invitation, and in the midnight, he's delivering a lecture for our students. Thank you very much, sir. Life sciences students are thinking that there are less opportunities to grow in life sciences field. In this scenario, I appreciate organizers for conducting this wonderful session on career opportunities in life sciences. Thank you, Pradyuta. Thank you, sir.
Thank you, madam. It's time to know about our today's speaker, Dr. Saber Hussain. I request Dr. Prasunna Solomon to introduce today's resource person.
Good morning, everyone. Today's esteemed guest is Dr. Saber Hussain. Dr. Saber Hussain is a senior scientist and professor of pharmacology and toxicology at Wright State School of Medicine in Dayton, Ohio. Dr. Hussain began his career as a research associate at IICT Hyderabad and then went on to work in prestigious research fellowships at Italy, Switzerland, and US. His research work on interaction of engineered nanomaterials with biological systems is well known and used in modern applications such as gene editing, mitochondrial specific cytokine fingerprints, organ on chip technology, bioimaging, and biosensing applications. He is currently the Associate Editor of Toxicological Sciences and a Fellow of the US Air Force Research Laboratory. He has won numerous scientific awards and has mentored more than 100 students who have since become scientists, professors, and industry professionals. His publications in nanomaterial toxicity are cited as must review articles and are among the most read papers in that field. He played an important role in identifying research gaps and rendering new technical skills sets. He is an expert reviewer of several government and private organizations. It is a great privilege for me to welcome my MSc classmate, Dr. Saber Hussain. We have done our MSc in Kakatiya University in the year 1986. Now, don't ask me, ma'am, why did you not rise to that level as him? Because by the end of this session, you will understand why. And I don't want you to make the same mistakes which we have made in our generation. You have so many opportunities, and the students must tell what mistakes we made. And we don't want you to make such mistakes. And that is the reason we have requested Dr. Saber to come and share with you what are the innumerable opportunities you as BZC students are having. And I want all of you to listen, concentrate, and then make use of these opportunities. That will be the outcome of this seminar that we are arranging. I want to thank you, sir. And I'm glad to invite Dr. Saber and introduce him to the students. And it is over to you, Dr. Saber Hussain.
Thank you, Dr. Prasunna, for your kind introduction. And I really thank you so much, Dr. Praditya, me the invitation through emails. And really, it's a great honor for me that to really give some my recommendations, my suggestions, share my experience. It taking me back to my BSc time when I was a BSc student. So it really reviving my past memories. So I hope, let me share my slides today. One second.
Yes, sir. 1 minute.
Can everybody see my slides?
Yes, sir.
Okay. So okay, I start the video. I don't know people can see me or not, but
Yes, sir. We can see you, sir.
Okay, thank you. So let me, okay, the presentation title that I have here, the career opportunities in life sciences. So this is a brief kind of a video you can see about a minute about the Wright State University, where it is located. It is a small town in the state of Ohio. It's a really a small, it's a reasonably well universities with the engineering and the School of Medicine and biology and math and a variety of disciplines. It goes like just maybe less than a minute and then I will talk. Well, this is the overview of my presentation today. I'm going to focus more on the career advice and passion in life science. What are the career options in life sciences? And what kind of skill and experience that we need to have to develop our career in life science? I would talk about few recent discoveries in life sciences because a lot of times that we think life sciences is very limited scope, but I will try to give few examples that were the recent discoveries in life sciences now. But at the same time, I will give my journey in life sciences. Prasoona is very particular about to share my experience, and I would be happy to share. Usually I don't talk about myself, but if it helps the student, I would be happy to talk about it. Then I will talk about the Wright State University. I have asked my colleague, who is a program director of admission into pharmacology and toxicology master program, because it's almost like over 10:30 p.m. here. I don't want to bother her, but she recorded like 5 minutes to 6 minutes, and I will share that information. She will give the overview, what kind of, the strategy you should have in order to get admission into the master program if you are interested. And then, of course, I will open this session for the questions. Just to disclaimer, the whatever the opinions I am expressing here today, they are really strictly of my own. They do not necessarily opinion of the university or the organization that I belong. I just want to make it clear that it is my experience and I'm sharing some of my advice to you. Well, let me start with this because most of you are Bsc students. These are some of the questions. I'm sure you have a lot of questions in your mind, but probably the most of the question generally you have in your mind, why am I doing BSc with the Bsc? What I'm going to do after BSc? Are there any potential to make career being in life sciences? So these are general questions, and I really don't blame you. You probably getting these questions by yourself, or you are getting by talking to the people, or you are getting sometime by the society and so on and so forth. But I try to give you some overview. Hopefully that will answer to some of these questions, and I'm sure you will have more. But if you do, we will discuss at the end. I want to make sure that you look at this article. It just came this year, actually. It's a recent one. And it's a nice overview about life sciences 2020. The future is here. The message is that you have seen so many discoveries in life sciences, the rapid technology advances over the past few decades, and there are enormous opportunities in life sciences. Actually, if you read this article, I'm not going to go in details. However, it is only limited to the US, what the opportunities and the employments for the past few years, frankly speaking, most of the opportunities in life sciences are significantly increased. And you will realize that at the end of my talk, because there are so many discoveries and the expansion of the life sciences into variety of other fields. And there is a combination and multidisciplinary opportunities. You being a life sciences student, you probably will have plenty of opportunities. And in order to accomplish that, and I will give you some advices. This is my advice to start. So before I talk about the discoveries and the importance of the life sciences, I really want to spend some time about my recommendations and some career advice. And these are very, very important for your career. The first is the stay focused and the time management. And this is really very hard for the students, particularly staying focused, because I know how you get some of the questions. Even if you talk to somebody, let us say you're talking to someone, they will say, what are you doing? The Bsc, Bsc, the first question you probably get, why you didn't try for other things? Why you are not trying? Why you are not trying that and this? It's nothing wrong with that. But really, regardless whatever they say, the focus is very important for you, what you are doing, focus and manage your time. And the time management is really very important. We only have 24 hours. I wish we could have more than 24 hours so that we can spend more time in the WhatsApp, but there is only limit for 24 hours. So it is really manage your time. I mean, look at your time, how much you have, 24 hours, and then you can look at what the time that you required uh, to do your other things, and how much time you can actually contribute for the study or for your career. That is really very important. The second thing, the more important, the willingness to learn new things. It's very important, because when you are a student, it's a general tendency that we want to study only for exams. And the strategy we have as a students, it's a common phenomena that we like to look at what the questions that they are asking the exams, and then we try to rush and try to prepare just before the exams. That is not the right strategy. Apart from what you are learning from your subject, and try to draw the conclusion from whatever you are learning in your class, the new things connected to that particular topics, and it is very important. The third thing, the self-confidence. And in particularly for the Bsc students, you need to develop the self-confidence. That's very important. I'm going to tell you one thing. Every individual has uniqueness in it. So you have certain unique thing in you that you have to identify. Please bring that up. Nobody is bring it for you. You have to identify yourself and you bring it up. You have some certain uniqueness. It may be hidden, you are not recognizing that. And then you need to bring it up and boost your confidence that whatever you are planned to do, you will do it. And that's really the self-confidence. And the next thing, the awareness of recent discoveries. If you are really the Bsc students, and make sure that you know the recent discoveries, what is happening recently, what happened, what are the new discoveries, where we are going in the next. That's really another thing. The next thing is the right mentor. I'm sure you have outstanding professors, lecturers, that they are trying to help you. That's the reason they organize this, to help you. And find the right mentor and really take their advice. And it is really very important. and then develop the critical thinking from that mentoring. And the last thing, have the vision. I mean, I will explain you what is the vision. I mean, you can have like a lot of dreams. That's nothing wrong with that. But don't select some of those and really stick to one and set up the goal. And when you set up the goal, your clock starts. And when the more important thing is how you execute your goals. And these are the things that what I said, the time management, the focus, willingness to learn new things, self-confidence, awareness, the right mentor, all those things will help you to down select your goal and stick to that and execute the plan and move on your plan. Like just make sure that you have really planned well, like step 1, step 2, step 3, step 4. And then when you achieve the step 1, that is your first milestone that you accomplished. And then when you achieve the step 2, that's your second milestone. Maybe you have like a 10 milestones to accomplish the goal that you are looking for. Let us say you want to be a professor or you want to be a lecturer. Like, let us have like a plan in your mind, whether you want to change your career from here to look for other options. That's fine, nothing wrong with that, but have your goal set and then work on executing those steps. And sometime I'm just telling you, when you are going through those steps, there is a good chance that you may fail in those steps in between. But if you fail, don't discourage by that failure. And every time you fail, you learn a lesson. And really don't ask why I failed. Ask a question, what I have done, what I need to do further. Because when you keep asking yourself why, then it takes you behind. But if you ask what, it takes you move forward. And there may be another way, but definitely you will have the right option that you will come up. Another important point here, this is my advice. Don't be distracted. That's on the left side. I listed, right side, I have listed few things. Don't be distracted when you are doing work, and don't try to think multiple career options. There is nothing like that. You can only do one. It is okay to think, but you must down select. But don't swim around with like hundreds of options. You're saying you're talking to one person, he's telling you something, you're talking to the another person, he's telling you something. But really you need to stick your what uniqueness you have, what the strength you have, and look at all those things and then try to focus on that, avoid the distraction. And sometime, as I said, if the results are not according to your expectations, ignore them, move forward and plan well. And it's a biggest mistake that when you are a student, you are disconnected with the scientific discoveries. Always try to read some scientific magazines, or there are a lot of other things apart from your subject. Read some of those, then you know what are the new things coming. Very important things, the negative advice. You know, a lot of people, when you talk to them, some of them, they really give you the good advice, but don't expect everybody gives you the same advice. A lot of times the negative advice, be careful with that. And a lot of times you have that kind of things. Please try to avoid those and move forward. And one of the strategy, as I said, studying just before the exam is not a good practice, and plan it well, way ahead. And that is the best strategy. These are some of my advices. I hope it helps. And moreover, be healthy. And I know everybody has a stress. Being a student, it's a lot of stress, but try to have a balanced diet. I mean, healthy diet. I'm not saying eat more, but I'm saying balanced diet. And sometime the way we will habituated in this modern world, looking at the snacks and the junk food, that's not good. And there are really very good studies that the students who have poor eating habits, there is a good correlation with the lower grades, and then they are susceptible to illness, that increase the fatigue, you get tired. And those are the things very important. Develop the balance with the nutritional. And more important thing is just be happy and relax. Regular moderate exercise would help. So look at that. I mean, there are really good studies that just enjoy the life, but make sure you focus what you're trying to do. Okay, what is the passion in life science? I mean, I'm just giving you certain advice that in the bottom one, the foundation, you already crossed that stage. Congratulations, because you have, if you are in a Bsc Bachelor of Science, you are on the second stage. You already accomplished one important milestone, that your foundation in science is pretty strong. So that is, you should be proud of that. Don't take that as a negative. Don't think that everybody does that, and think that that is your first milestone that you accomplished. Now you are embark on a journey. You are on the second step. So this is a great milestone for you. This is your second milestone. You need to work on that. So, and what I'm trying to do here, just to give a very broad kind of a overview, when you are in the BSc or the BSc, and it is very important that you need to have the goal. And frankly, the students, particularly in the first year, they have really a good amount of time, and they have to be very careful how they manage their time, how they set their goals. And if you really want to do, I mean, there are several options here. I mean, you have options to think about in industry, in the academic, in the non-academic. And most of the time, like if you want to go all the way up to the academic, the skill experience, like for example, you want to be a lecturer or the professor, the principal investigator, or maybe the research scientist or the study director, maybe you should be in the clinical research. There are plenty of opportunities, but in order to go there, in order to accomplish that, you need to go through these steps, like the bachelor, master, doctorate, and then you also need to have a postdoctorate training after your PhD. So if really this is your passion, this is the right way you should set the goals. Well, I'm really more like an industry. Maybe you are good in the lab. You're probably very smart in the experiments, and you enjoy being in the lab, but you're also interested in the applied science. Maybe you try to look at the industries and think about that, what the skill set you need to develop. And there are plenty of labs that they will take as a research assistants, research associate, and the biologist, and there are plenty of things that you can take an advantage. Now, there are other things that non-academic, that, you know, you are in the Bachelor of Science, you are doing the BSc, you are studying the science, you're also studying English, and I'm sure you have some introduction to the computer science. But perhaps some students, maybe they are very gifted, they might write well, they enjoy writing, but you know, there are plenty of opportunities as a writer. I mean, every pharmaceutical industry, they are looking for a good scientific writer. What they do, they read all the journals, and they read the whole literature about particular drug, and they summarize it and they give a report. So that is a very good skill set, don't underestimate. And that is also a good skill set to develop. And maybe perhaps, you know, like you want to go into an administration in a other area completely. That's fine, nothing wrong with that. Whatever you have in mind, set the goal and work for that. And you will be surprised that some people, like, they do master and they do PhD, and I have seen several of people after PhD, they are really some kind of, they went into this non-academic type of environment as a patent attorney, like, you know, like a editorial, in the journal editorial, biostatisticians, and the technology transfer, and the patent. The patent is like when they have a PhD and they go to do the law, and they specialized in the patent. The patent means when you do research, when you discover something new, that is your intellectual property, and you want to protect it. So what the patent specialist does, they will take all the information from you, and he will make a good story, and then he will review it. So it's a very good profession. So there are plenty of opportunities that you can think of while you are a student to consider some of the options, but make sure that don't go to these because somebody is asking you to do. And that is the biggest problem, because your parents want you to do something or your friends want you to do something, and that strategy is not good. You have to think whether you have the passion, whether you have certain strength to accomplish this task, because you know your strength and weakness. Nobody knows exactly as much as you know. And just smartly plan your career. There are plenty of opportunities. That's probably my best message. If you want to go for like master and the doctorate and postdoctorate, if you want a mentorship, please contact me or contact anybody. They will give you the advice. And myself, I'm really more in the academic. I would be happy to give more academic advice anytime. You're most welcome to contact me. What is life science? I mean, I'm just trying to give you some kind of a very brief introduction. I mean, the life science, historically, you will start studying from a basic gross organism. Like, you study the structure of the organism and the structure of the tissue level. You probably study the digestive system, respiratory system, all those at the organ level. But really, that's not limited life sciences to that part. You need to really think from a different angle. Like you can go further, like organs, tissue, cells, molecules, atoms. I mean, all those things, when you put together all these pieces, really you are going into in a systematic scientific study at the molecular level. What is happening, how the process, the cell communicating each other, and then you're going into organism specificity to universality. So you can see how the whole, in the mid portion, you are studying a little bit more broadly about the organisms and the tissue and the cell. But really, when you go to the cellular level, you're going even the deeper into the molecular level. What are the genes? How the genes storing the information? How it is processing? How it is executing? How the cells sensing that and then taking and communicating to the other cells? And how it's the whole system of biology is like a computer. It's a great systematic machinery. So that is where you have to think. The life sciences is not just limited to one. And now you can think about your career, how much wide scope is available when you look at the whole picture. And I will give you some examples, some more examples. Let us say that this is really some kind of like an overview of some of the areas to catch your interest. That the life sciences is nothing limited to botany, zoology, and chemistry, but it is beyond that. Come out of that like a thinking that if you go to one step ahead, it is little bit more deeper, that you are going biochemistry, genetics, immunology, that historically you probably have this subject attached in your syllabus one way or other way. I'm sure you are studying very briefly or at least little deeper introductory topics of these. And then microbiology, biotechnology, and the stem cell biology, and all those things are the areas they are connected in life sciences. Sometimes they may have like a special the courses, but ultimately, being a Bsc student, what I'm trying to say that you are getting to introduce to these areas if you really think carefully. I mean, like neuroscience, pharmacology, physiology, and all those introduction indirectly or directly you are covered in your syllabus, if you think about that. And now the another area, the environmental biology and the toxicology, looking at the toxic effects of pollutants and the toxic effects of variety of pesticides, the chemicals, and every day that in this modern globalization, industrialization, there are thousands of chemicals coming out. And really the system biological impact of those chemicals and the biological impact, environmental impact of those chemicals, it's a huge field. I mean, I'm sure that in one way or other way, you're studying those in the form of ecology or in the form of environmental biology there is always these topics are included. Even when, like 25 years ago, when I was a BSc student, I have these topics one way or other way covered in the whole syllabus. Now, the more important thing that I want to say on the right side, and I will talk to you very briefly about this little bit, gene editing technology. So I will talk about that very briefly because you probably heard about just about a month ago, there was excited news that the Nobel Prize for this year in chemistry, who actually did a lot of work in the life sciences on the gene technology. And I will talk in the next slide what they have done very briefly. And the another one, microRNA. It's a non-coding genes. They are coming into the play a very important role that, like, read about those and think about. There are several pharmaceutical industries. They are very interested to identify microRNA as a markers or the biomarkers for a variety of diseases. And that is one of the very wide open area for research. And it is really coming out pretty huge if you look at in the literature. The another thing is the quantum biology. And I will give you a few examples about quantum biology, what it is. Synthetic biology, I'm sure you heard about synthetic biology, system biology, the microbiome. That is really becoming more popular in medicine now because most of our gut microbiome, the gut bacteria in our body, they are all beneficial, friendly bacteria. They're actually playing very important role in the signaling and giving information to the brain. There is a lot of actual information and the opportunities right now. If you look at that, you can even buy some microbiome in the form of pills that the people will take, like, like the bacteria that you will find in the yogurt. So that is where now they are incorporating these very beneficial microbiomes or the bacteria to help, the boosting our immunity and all those things. It's a wide area right now. And the another one is the optogenetics. Like, how can you activate the gene by light? How can you trigger the brain cells by the light? I mean, this is very exciting topic that people are working. The radiogenetics, like radio frequency. How can you trigger the genes by applying the radio frequency? And this is another one. Epigenetics. I mean, epigenetics is probably you must have already learned in your class the methylation of the DNA and slight, like the change, like if you take any kind of a nucleotide, if it is replaced, the hydrogen replaced by the methyl group, and then what happened, the sequence of the DNA is not going to change, but the functionality of DNA is going to change. So because of all these like modern chemicals and all those things, there are so many topics that are coming up in epigenetics, and that's another area. Then other area is the bioprinting, 3D bioprinting and the organ on chip. So that's again, I wish I could have time to talk all those things, but you will be really excited to learn about organ on chip. What is organ on chip means that they are fabricating the tissue and the organs by using the 3D bioprinting in order to recapitulate how the organs are actually growing in the in vivo system. It's another exciting technology. And imagine that the most of those skill set that they required from the life sciences, that you are spending the whole time in the life sciences studying, then you will be the perfect candidate to contribute to these areas. Well, just recently the Nobel Prize won the Dr. Charpentier from France and Dr. Jennifer Doudna from the University of California. They are the one who just got the Nobel Prize for their 2 decades of work for the CRISPR-Cas9. Just to give a brief introduction, the CRISPR-Cas9 is one of the exciting technology. 10 years back, the scientists actually discovered that in bacteria. The bacteria, what it is doing, they are, when they infected with the virus, viral genetic material, when it is introduced into the bacteria, and they found in the bacterial gene, the bacterial gene is very small, right? I mean, we all know. So they have some of the systemic the Cas9, the pieces there connected with the spacers in the bacterial gene, what it is doing, as soon as it sends the foreign, the genetic material from the virus, it actually induces the transcription in order to bind to that the genetic material, and actually it degrade the viral, the genetic viral, the gene. So what happened by doing that, the viral get inactivated. So indirectly, the bacteria has its own immune system. It is protecting from the virus. So the scientists actually took this, like the whole phenomena, and they give a deep thought. Why don't you take this machinery or the phenomena, why can't we introduce into the mammalian cells? And they have succeeded that. And now they have succeeded this, and they took this, like the DNA, they introduced to the guided RNA to that, and then they're surrounded by the endonuclease protein. You can see the how it is, and then they introduce the whole thing into the mammalian cells if they want to inactivate particular gene, or if they want to, correct particular gene. And for that revolutionary research, they actually got the Nobel Prize, and it is going really very popular now because it is going to be applied in the medicine to correct a lot of diseases by inactivating the genes or correcting the genes. So there is a large scope in that. If you look at in the literature, there are so many companies that are actually working and developing this technology and taking and transitioning this to the clinical application. So there is a lot of scope in that in research. MicroRNA, I don't want to go in detail, but just in a nutshell, this microRNA is a small non-coding genes. They're coming out of the nucleus. They are very, very small, like about 22 to 25 nucleotide, and there is a whole sequence of the process, how it is processed by nucleases, all the way coming into the cytoplasm. But what it does primarily, it is silencing the gene. So it is taking very important. So most of the stress situation that we have, there are certain situations that the microRNA very specific to those, and those microRNA are serving very good biomarkers. For example, for cancer, for other diseases, they are identifying very unique specific microRNA. It is a very detecting tool to identify some diagnostic and for therapeutic interventions. This is another quantum biology. I mean, you are a biology or the Bsc student, and you probably heard about these birds. It's a European robin bird. What it does, it's actually migrate from southern part of the Europe to the northern part of the Europe in a precise manner by following the navigation. So the scientists, they discovered that they have a magnetoreceptors, and after that, it has become really a popular area of research, because if you look at the whole photosynthesis, how the photons, the light hit some of these compounds, some of these molecules in the chloroplast, and then it actually introduce the photons to that molecules, and through that photons, the electrons get activated, and then it actually have some energy, and that energy being transferred, and then it is giving in the form of ATP. So this is the whole quantum process. So it means that they are really following the principle of quantum physics, and you as a biology, you better you understand actually more what is happening into the biology. So you are not studying the physics, but you are looking the effect of quantum effects on the biology. I mean, these are the very good opportunities, and there is a large scope of research in the magnetoreceptors and understanding the entanglement of the photons, like when the electrons, they spin over in opposite side, and how the energy being transferred throughout the biological system. And that is one of the remarkable research which is going on. You have plenty of opportunities over there, and I'm sure you have studied some of the quantum uh, properties in chemistry, in the physical chemistry, and you probably studied in the physics as well in the high school or in the intermediate. But the same very simple principle that biology is utilizing to make the whole process of the signaling. The next thing is the microbiome. The microbiome, very quickly, that our gut, the gut has like a millions of bacteria, and most of these bacteria back in early 90s, and they started identifying these bacteria, what kind of specific role these bacteria has. And you will be surprised knowing how this bacteria is playing a role in the human system, uh, how essential it is. And slight modification of this gut bacteria really trigger the dysfunction of some of the organs. I mean, there are like a huge studies, and it was like recently in the Garden News, like it was pretty much like they were they kind of described the whole importance of microbiome and where we are going and how valuable this research for the clinical application. Well, the another area is the bioinformatics. I mean, all those areas when I'm talking, keep it in mind, there are so many areas that I'm not talking, I'm only talking very few. This bioinformatics is another area because some of you might be very good in the computer. You are really good in biology, in the chemistry, but also you have the passion in computer science. But at that point, you can combine your computer science skills. If you have a little knowledge about the statistics, you can apply that. And you are very strong in biology anyway. When you combine this, you are developing a new field about bioinformatics. And it is a huge and very good market for the bioinformatics now because of machine learning and the artificial intelligence. And these are the two programs really taking a lead now, and there is a large scope of biologists to taking the process of information, what is happening in the cell, all the gene sequencing, protein sequencing, and all those things, and apply into these bioinformatics to really create the virtual cell. It's a predictive cell. Then you can really predict whatever happen in the cell, you can predict in the computer. In other words, the computer you are making as a human system. You are really developing the system by using all this knowledge. So it's a really great field. And some of those, if you want to more information to get general sense, these are some of the key websites I have here. What you could do, you could go there. And I know if you are BSc first year, second year, you might not be there to apply, but that's not the purpose. But at least you get the general sense. What kind of jobs are there? What kind of skill sets they are looking for? And try to look at that. I mean, try to see, then you get really a general kind of a knowledge, and then act on it. Like, okay, this is my requirement, I'm in the first year, what should I do? Maybe I'm interested in research. Why don't I go to some research labs? Like in Hyderabad, there are so many research labs like IICT, CCMB, NIN, there is a NIPER. Maybe look at that. There is a student summer opportunity internship. I mean, in US, every university or every research organization, they take undergraduate students for the summer internship to train them for science. So those are the good opportunities. Look at those, then you get some networking, you know the people, you know the opportunities, and like expand your scope. I mean, because you have like a plenty of time really. And people always think, well, when I go, I don't have time, but you have the summer 3 months time. I mean, 2 or 3 months time, take advantage of those to learn some additional skills related to your passion. So with that, I think I tried to give you like a very kind of overview. I spent a little more time to give you kind of advice and some encouragement. Look, how many opportunities you have. There are so many areas. There are so many opportunities. You can take advantage of those opportunities and read little bit more information. If you are interested in a particular topic, I would be happy to talk little in depth, in more detail about those topics. But I gave you really a very kind of a general overview. Let us talk about my journey very briefly. I will go very quickly here. This is briefly that I am common, like average student like yourself. And I did BSc back in 1980 to 83. And I did my master in zoology and in Kakatiya University. and Dr. Fasuna Soleiman, she's my classmate. That's how I came to know about this seminar, and thank you very much for inviting. I really enjoying talking to you guys. And then I did PhD in Indian Institute of Chemical Technology. And I'm just telling you that this is my career change. So I'm just telling you that the BSc and MSc, these are really turning point in your life. So if you really take advantage of that time and the focus, because otherwise, once the time is passed, it is hard to reverse the time. So take advantage of that. And I got the honor to be in Indian Institute of Chemical Technology. I did my PhD. It's 3.5 years. And after that, I kind of I had in my mind that I need to be in academic. So the opportunity came and I tried to, in between, after my master, actually, I did some 6 months for experience in AV College, which is still in Gagan Mahal. And I actually spent there 6 months. And imagine, it's a really great experience for me. Right after MSc, there was an opportunity, and I enjoyed working with the senior lecturers, and it was a great experience for me. And then PhD, during that PhD, I got the honor to attend the international symposium in Italy in 1990 for 2 weeks. And that is really the right time for me, because for the networking, that is where I got a lot of exposure. And I was very fortunate because they only selected like a 30 students, 15 from their own country, from Italy, 15 students from the whole international. I was actually very fortunate to selected from India, because they paid everything, like a boarding and lodging. It was very competitive. You had to write a research project as a student for 2 weeks. What will you do there? And I wrote a project on metallothionein, and they accepted it. I was very fortunate. The second, that right after my PhD, I was very fortunate to get like a very short-term research associate in the University of Urbino in Italy. I spent about not exactly 1 year, it's about 9 months to 10 months. And after that, I got really a postdoctoral fellowship in the Geneva University Medical School. And I will tell you the brief experience. I mean, while I'm showing that, the one thing I would like to tell you, that I started with botany, zoology, chemistry, and then zoology as a MSc, but then partly toxicology. And I really, by the time I went to my postdoctoral, my research was completely different. I mean, it's totally different than what I have been doing. I mean, that's where I want to give you that when you are doing one thing like a botany, zoology, trust me, you are not limited to that. You are not at all limited to that. There are scopes that you can expand your knowledge deeper and deeper and move on. That is possible. And then after that, I was like a visiting scientist in a FDA lab in Jefferson, Arkansas in USA, 94 to 97. That is again very honored to get that. And after that, I was assistant professor in veterinary school of medicine in Tuskegee University, Alabama, USA, maybe about less than 2 years. And then I finally came here to Wright State back in 1999, because this was a great opportunity, and I came. And since then, I am here. I'm really enjoying working with Wright State, and also I am connected to the Air Force Research Lab. I have a nice projects with giant projects, and I'm really enjoying with a lot of students. This is my PhD talk. Whenever we do research like that, that's my recommendation to you as well. As a student in the class, you attend the class, the professor, ma'am or sir that giving you the lecture. But after lecture, don't just limit to the lecture, like think in a deeper, when you heard, for example, something in that lecture, and ask some questions to yourself, that what is next, and always think like in a big picture, going to the deeper. So in research, always it starts with a question. What is the question here? The question was whether the bioaccumulation of heavy metals affect the normal function of the organism. The heavy metals like mercury, arsenic, lead, and cadmium. So these are the toxic metals that when it is accumulated in the body, our body is not prepared to handle some of these. And some of these, yes, they induce the proteins and they bound to them. And some of the metals we cannot. So the question is when they go into the body, how their body respond. Then I took very simple model in Indian Institute of Chemical Technology, working with Dr. Kaiser Jamil at the time. She's now retired. So the question is to grow this water hyacinth and take these very, very unique phytophagous insects. They particularly feed and grow only on these plants, and feed the plants with these heavy metals and see how much it's accumulated in the roots and the leaves and the different parts of the plant, and then measure how much it's actually accumulated in the insects when they feed onto these. So what I did, I looked at some very unique biochemical parameters of these insects, and I looked at some kind of the expression of new stress proteins in response to these, and we used a lot of electrophoresis at the time to identify some unique proteins. And the bottom line is, during my PhD, I really tried to look at the biotransfer of metals from the plant system to the insect system as a model for the food chain, because everything is going through the food chain all the way up to the human being, right? And that this is one of the model system that we took to study the impact of these metal compounds in the organism. And this word biotransfer of metals, and that is the one which was very catchy. I think that's where probably worked for me, going into the next direction, to apply my basic question to the mammalian system. That's how I got in Europe, and where I work in a completely different model. I mean, I work in the insects and the plants, but when I went to the Italy at the University of Urbino, this topic was totally different. I was studying on the metal binding protein, the metallothionein, and again the question was about the metals. Like when you expose the cells, these are the CHO cells, expose the cells to oxidative stress and the metals, how that the resistance, how does the cells resist from this exposure to these metals. And one of the protein is metal binding protein, is a metallothionein. And I really studied on these cells and isolated some clones and look at the gene regulation and identify these proteins unique to oxidative stress in when the cells exposed to hydrogen peroxide, and some of these research we did. And these are the two papers we published. Finally, when I went to the Geneva in the Switzerland, the research is completely different. Now it is, the concept is the same, but the model is different. And now I'm working on a human lymphocytes because it's a medical school. There is a clinical application, and the liposomes. And these are the lipid molecules. And we try to introduce some of the novel compounds that I prepared. I learned a lot of chemistry over there, like a cysteine and silver cysteine. When you combine these two, how the mixture of the compound and what is happening here, how the functional groups are sharing between silver and cysteine, how it is interacting with the enzymes. When you incorporate into the liposomes, can we introduce into the lymphocytes? If the lymphocyte is infected by the virus, ultimately, can we use these metals such as cadmium, silver, and zinc as a potential antiviral agents? That was the question. And we did really good job, like in 3 years, and we published like a 6 papers. So that was really another move for me to go from Europe to USA. And when I came to USA, I'm not talking what I did in a FDA lab or in the university. It probably takes more time, but I just want to give very little introduction what my current projects are. My current projects are related more in the nanotechnology, and it is completely different now again. I mean, I had to learn about nanotechnology, the characterization of engineer nanomaterials, and so on and so forth. So it's a really a lot of good experience, but I'm really enjoying. And the question is, how does this engineer nanomaterials interact with the living cells? It's a simple question. In order to address that, some of these tools, that gene editing, the dynamics of mitochondria, cell signaling, and some of the multicellular model that we are developing, and we are also working on the organ on chip, and then this bioimaging, biosensing, because that's the whole essence of this project. And we use a lot of electron microscopy and looking at the uptake of these particles in the cell. And also we look at the signaling and how these engineer nanoparticles interact with the cells, how the membrane potential of mitochondria change, and as a result of that, and how it impact the whole cell functionality. This is very briefly about nanotechnology. If you think about like the football size, and then you can come here, and these are the material in the nano size. Just in a very quickly to tell you, you take 1 meter and divide it 1 billion times, not million, billion. So that is the small about nano size or the nanometer, and that is where we are dealing with this. So this is like a, right now we are dealing with the 4th generation of this nanotechnology for the molecular devices, atomic design, emerging function. It's a huge field, and the nanotechnology itself is a good lecture, and I don't think that I can talk much on that, but it is just to give you the brief introduction about what is this nanotechnology. They are really extraordinary applications, like in the personal clothing, entertainment, cosmetics, like all the cosmetics, sunscreens, medical devices, all the coating, energy, automotive, the cell phones, all those things, they all with the nanomaterials. And some of those, look at here, basically the single wall carbon nanotubes, multi-wall carbon nanotubes, the graphene and the fullerene. The one who discovered the fullerene back in 1990 from the Rice University, he got the Nobel Prize. And the one who got like this graphene and the carbon nanotubes, and 3 years ago, they also got the Nobel Prize. So this is the very hot topic, and there is a lot of scopes in the nanotechnology, nanobiotechnology. And this is very simple, that I just draw the cell structure just to give you very quickly, because you all know the cell structure. And some of the fundamental questions, how these particles interact with the membrane, how they are going inside, how it's interacting with the cell organelle, how it is triggering the cell signaling. The whole point of doing this, really to looking at when the particles go into the nano size, their surface area different. They don't follow classical physics, they follow the quantum physics properties, and they follow certain unique properties. So the question is, can our cell interact with that, and they have some kind of a biological interaction? If they do, how our cell respond to those unique, completely new particles the cell never even seen before. So the whole mechanism we try to investigate, and this is really to give very brief kind of a the whole mechanism of the stress, because anything the cell senses, like any kind of like a stress, it could be physiological stress, it could be the chemical exposure stress, or maybe you're working like too hard, and any type of stress, it's really induce some kind of like a antioxidant capability, because our cell has the capability to completely deal with the stress. That's the phase one. But if the stress keep on increasing, this is where you increase the pro-inflammatory responses, like interleukin 1, interleukin 2, interleukin 3, and then TNF alpha, that's tumor necrosis factors, and all those things like pro-inflammatory responses, and those are the cytokines. And then then it trigger a lot of signaling, like MAP kinase and all those, the complex things in the cell. And then you can really, if the stress keep on going, and ultimately the cell goes into necrosis or apoptosis, finally it's the cell death. So this is really the whole response, how the stress trigger in the physiological response in the cells. That is where we are trying to look deeper in the cell signaling. This is just to show that the electron microscopy, how we do routinely. I mean, we take the cells, we culture them, we process them, we use the microtome, and we look at in the microscope, and then look at the cells. Because this nanostructure material, you cannot look at in the normal microscope. You have to have the electron microscope. So that's why we visualize these materials in the cells, and we figure it out if these cells are really happy with these materials. And then you can see that whether we can develop the biosensors or imaging from these materials. So it's kind of like a very interesting to see. And this is some of my research that I looked at all these, like in particularly the gold nanoparticles in the 30 and 60 nanometer, how the cell respond to that. I don't think that I can go much on these, but what I'm trying to give you the whole comprehensive, the gene analysis we did, then we kind of identified some unique pathway based on these particles. You can see all these pathway, how it is analysis. Just to conclude here, that the depends on the size of these particles, the cell actually sensing these particles and acting based on the size and aggregation of these particles. You can see all these inflammatory genes and most of the signaling, and it's a very packed material. So it's really, we try to identify some of the unique the genes compared to the unique pattern, like pattern of these particles. So I wish I could talk to you a little bit more how the structure, the cells are sensing, how we can detect them. We have very nice electron microscopy pictures, but I'm not showing because of the time. And all these actually published in these papers. So if you look at these, it is available on the literature on the CRISPR-Cas9, and looking at some nice the silicon nanowires, how the neuronal cells actually interact with that. These are the astrocytes, and you can see how we recorded the whole electrophysiology of the cells. And some of those, like we developed some of the in vitro aerosol exposure, and we also looked at very deeply in the mitochondria on the Golgi complex when they interact with these nano size materials. And one of the things that one paper that we actually published in Nature, the whole protocol, how to visualize these particles in the cells. And this is something that I have, like the Wright State University has a master program, and you can go and look at in the website. And I asked Dr. Terry Orzi, she actually uh, recorded one. So there is only 6 minutes. You can see I almost done. If you have like a 5 minutes, look at this video very quickly. It takes less than 4 or 5 minutes.
Greetings. I am Dr. Terry Orzi. I am the Director of Education and Assistant Professor at the Boonshoff School of Medicine, Pharmacology and Toxicology Department. Dr. Saber Hussain has asked me to take a video and condense it down so he can share it with you today. If you want to see, skip a few of the items and go straight to the aerial view of our beautiful campus. So that structure there is the double helix. Okay, now I'm going to take you over here to our folder so that I can introduce you to the team, give you a briefing on the timeline, tracks, and concentrations. So let's get started. So this is Dr. Jeffrey Travers, and he is our chair, and he's a professor, dermatologist, MD, PhD. This is me, Dr. Terry Orzi, and this is Miss Catherine Winslow. This is Corinne, she's our program coordinator. And this is Barbara, she is our admin specialist. If you apply to our program, you're likely going to speak to one of us. Just give you a brief history of our program here. 2001, we started the MS research track. In 2009, we started a non-thesis track. Our space was limited, so this was our best option. 2012, we created the CBRN certificate program. And in 2014, we rebranded the non-thesis track to be a leader admin track. We really wanted to pull in some courses from business, leader, and administration.
Oh, okay.
In 2015, we created the MDMS Clinical Investigation Track. 2018, we created a one-year online track. In 2019, we redid our MDMS track. And in 2020, we created the Clinical Trials Coordination concentration. We are currently working on a one-semester certificate clinical trials coordination, as well as a healthcare and homeland security concentration. We have 4 tracks. We have a research track, 5 semesters, leader admin track, 4 semesters, leader admin track, 2 semesters, and a leader admin track online. We have 2 concentrations, the clinical investigation concentration for medical students and the clinical trials coordination. Now, the difference between a concentration and a track is the concentration will show up on your transcripts, the tracks will not. So we have several electives that you can take in addition to the required core classes. And if you take those electives, some of them will give you certificates. For example, we have department certificates. You see 3 here. We have the graduate certificates. Those are the CBRN certificate. And as I mentioned, we're starting a clinical trials certificate. And then the Six Sigma Green Belt and Black Belt certificates. So that's really a brief introduction to our program. Again, you can find more about us online, and I'm going to show you where you can go to get that information.
Oh, I'm sorry. Are you still not able to see this? I think we have a problem sharing, so I probably, I don't think you are seeing the video. Is it the video is visible now? Hello?
Yes. Yes, sir.
Oh, okay. Okay, so hold a second.
Briefing on the timeline,
see now?
tracks, and concentrations. So let's get started. So this is Dr. Jeffrey Travers, and he is our chair, and he's a professor, dermatologist, MD, PhD. This is me, Dr. Terry Orsi, and this is Miss Catherine Winslow. This is Corinne, she's our program coordinator. And this is Barbara, she is our admin specialist. If you apply to our program, you're likely going to speak to one of us. Just give you a brief history of our program here. 2001, we started the MS research track. In 2009, we started a non-thesis track. Our space was limited, so this was our best option. 2012, we created the CBRN certificate program. And in 2014, we rebranded the non-thesis track to be a leader admin track. We really wanted to pull in some courses from business, leader, and administration. In 2015, we created the MDMS Clinical Investigation track. 2018, we created a one-year online track. In 2019, we redid our MDMS track. And in 2020, we created the Clinical Trials Coordination concentration. We are currently working on a one-semester certificate clinical trials coordination, as well as a healthcare and homeland security concentration. We have 4 tracks. We have a research track, 5 semesters, leader admin track, 4 semesters, leader admin track, 2 semesters, and a leader admin track online. We have 2 concentrations, the clinical investigation concentration for medical students and the clinical trials coordination. Now, the difference between a concentration and a track is the concentration will show up on your transcripts, the tracks will not. So we have several electives that you can take in addition to the required core classes. And if you take those electives, some of them will give you certificates. For example, we have department certificates. You see 3 here. We have the graduate certificates. Those are the CBRN certificate. And as I mentioned, we're starting a clinical trials certificate. And then the Six Sigma Green Belt and Black Belt certificates. So that's really a brief introduction to our program. Again, you can find more about us online, and I'm going to show you where you can go to get that information right now. This is the Boonshoff School of Medicine webpage. I'm going to go to departments, scroll down to the academic departments, pharmacology and toxicology, educational programs. You can find out everything you need to know about the program, the department, the tracks, the concentrations, how to apply, what are required. For example, you see here, TOEFL score of 213, 79 IBT, and then on band 6 for the IELTS, a GPA 3.0. Additional information can be found here, which includes other things that you need to apply, like transcripts, letters of recommendation, personal statement, and where you apply. So you can just click here and apply. And if you scroll down a little bit more, you can see what the application process looks like and what you need to do. So it's really very simple. I just want to briefly come over here to faculty and staff and scroll down there. You see the chair, you see some of our other faculty. Take time to look, but right now we're going to focus on Dr. Hussain. This is Dr. Hussain's webpage, and you can see how many current students he has. His research focus, which includes, as you can see, quite a bit of nanomaterials. He also does molecular signaling. We're going to go back to his publications, and I just want you to see how you can apply to spend time with him as a master, doctoral student, postdoc, a research associate, etc. So let's look at his Google Scholar page. He has over 250 publications. The list keeps going. Check that out. Isn't that amazing? I remember when Dr. Hussain got promoted to full professor, people said after looking at his CV and qualifications, they should have a statue in the university square dedicated to him. That's how well respected he is in the university as well as at the base. Which is why I'm going to now turn this over to him so he can talk to you more about his research, and basically anything you might want to know, he can do that for you, answer some questions, etc.
Okay, so let me stop these. Okay, so now it's a time for questions. Sorry, it took a little bit time because of the video and all. I hope you enjoyed. If you have any specific questions, I would be happy to answer. Thank you very much. I wish you all the best.
Yes, thank you very much, sir. You really inspired all of us. As you have rightly said, everyone should have a uniqueness and should have a focus on time management, vision, and plan. Really, you have inspired not only students, your words and insights have inspired all the faculty also, sir. We all know that it's too late night for the US. It's really appreciable that you have made our day a wonderful day. Sir, we want to continue this association with you and with your university from now onwards, with your permission. And we also like to invite you to our college whenever you are in India, Hyderabad. Thank you, sir. Thank you very much.
Sure. I have some good interaction with the professors with the different universities, and I would be happy to ask them to talk about and share their experience. And in fact, I was actually thinking maybe to have some students to talk, but this is kind of not the right week because this is a Thanksgiving week. It's all the holiday season. Nobody is in the campus, and also it's kind of a little late, so I didn't tell, but I already talked to them. They are willing to volunteer even though it is late, but maybe sometime Whenever next time, I would be happy to do that. But for the students, please, you have my email, maybe in the slide, the last slide, you have my email here. I don't know whether I may have to share it again, but if you need to contact me, please don't hesitate at all. Please contact anytime to this email. So I'm just sharing this. I hope everybody is seeing this email. You're most welcome if you are interested in this university, because the degree master is in pharmacology and toxicology, the one she just talked about, but there are also other programs. That's master in biochemistry, master in biology. There are so many other programs, and you as a Bsc, they look for 4 years, but if you have 1 more year addition, maybe you are qualify for that after GRE and TOEFL. But if you need anything, or if you want to work on that, please contact me. I would be happy to help in any way I can.
Yes, sir. We like to open the session to students. Students, you can ask the questions.
Please.
Sir, here is a question. You talked about using heavy metals like silver and cadmium as potential antiviral agents when lymphocytes are attacked by virus. Was it really a potential antiviral agent at the end? Bhavika has asked this question.
It's a good question. I was talking about like back in 92 to 94, when I was doing that research. We were trying to at least have the silver in combination with cysteine, whether we could use it as an antiviral agent, and we published a paper. But to be honest with you, it is still at the research uh, level, so it has not come to the application part because of 2 reasons. One is because when you introduce the silver, the specificity is the issue, because when it goes to the lymphocytes, it's not only killing the virus, but it is also hurting the lymphocytes, right? So what we try to design, we try to combine that with the sulfhydryl group combination. We try to look at some of the, like a cysteine, N-acetylcysteine, and other type of sulfhydryl group proteins that we try to mix with that. Then it actually, what happened, the virus, for example, some of those HIV, for example, it has a TAT protein. The TAT protein has a cadmium. The cadmium is required for like a dimerization of the virus. So if you really take that cadmium out by taking some chelators, you can effectively target that virus. But the biggest problem that we had, it's a specificity issue. So to really answer your question, it is still at the
Guys, if you have any questions, please kindly post in the chat box so that sir can answer.
Yeah, I can look at the chart. So I think the same question that I can see this about silver and cadmium as a potential antiviral agents, I already answered. That was my research back in 92 to 94 when I was in Europe. If you have any other questions, please. Yeah, the 4 years degree is like, for example, if you are doing Bsc, they want like the undergraduation here in US normally 4 years. So you have to have 1 more year. Either you can take the diploma to complete the 4 years, it is possible, or you can do the master 2 years in India, but you can also apply like after your first year of master. So usually they take 4 years, but some exceptional cases, they might admit you after 3 years, but you have to complete more courses here because you don't have like the 4 years. Instead of doing 2 years master here, you might end up doing 3 years. But if you take the another track, because the pharmacology, they started the new track now, they're like a thesis, non-thesis. The non-thesis track master is for the leadership. So that's only like 1 year. They reduce the time so that the student, they just, they might not be interested in research. They might be interested in the leadership. So there is a 1 year option as well. But regardless, they prefer to have 4 years. The another question, whether the CRISPR-Cas9 is safe for treating the human. I don't say it is safe yet because it's a new technology. It has been, there is a lot of progress on it. There are always ethical issues. There is the safety is one of the concern. But if we really do smartly, you just have to weigh, because any drug you produce, for example, none of the drug is safe. You just have to balance whether the drug has the benefits versus risk. If the benefits are more, you apply with a great precaution. If the risk is more, then there is the question, that won't even come into the clinical side. So it just depends upon for what condition you are using, how much risk you can take, what are the balance between the risk and the benefits. So it's really hard to say whether it is safe at the moment, because this is like a new technology. Very, very few experiments have done on the real clinical side. There's a lot of things to be done, but there is a hope. So research on diabetes, what should we do after MSc? Well, like research on diabetes, there is a lot of research going on. There is you are working basically on the pancreatic cells. There is a lot of genetic engineering going on. There is a huge scope. If you look at the NIH website, you can find out there is a lot of, so there is one professor in pharmacology and toxicology. He does research on the diabetes. So I can connect you if you are interested. What should be the greatest strength of life science aspirant? What do you mean by greatest strength? The strength is, like, as I said, well, the strength, you have to develop the strength in the life sciences in the area that you are interested in. It's very hard to tell just one strength. I think we talked about a couple of slides about my advice. I think that would be great to take it. The controlled toxicity in humans and any other organism lead to mutations or evolution. Yeah, it depends on what type of toxic materials we are looking. There are a lot of materials, they do induce mutation, DNA damage, single strand damage, double strand damage. There are so many chemicals that they do it. The forensic science and biotechnology after BSc, I don't think, if you want to become a forensic specialist, like at least in United States, you have to have a board exam. I don't know about in India. I'm sure they probably have some kind of like a training that you need in order to work in the forensic lab. Actually, there is one forensic lab in Hyderabad. That's one way I would recommend you to start looking the summer opportunities as an internship, even volunteering. A lot of times learning is more important, because as a student, focus more on the learning than earning. So the earning will come later, but the learning is very important. So if you are interested in the forensic science, I would recommend, or in the biotechnology, in the applied biotechnology, I would recommend to do some internship in a good industry or a good research lab to get some experience while you are a student. So if you are interested in diabetic, maybe the best is to contact me. I will connect you to the people who are doing that kind of research. Can you please explain about different research routes for food and nutrition? There are a lot of research on the nutrition side. but not much on the right state, but there are other universities. Yes, the GR is recommended to do a master in USA. Most of the universities, they like taking that would be really good. It depends on which university you are going to. If you want to go to the top university, you need to get a good score. Microbiology, there are plenty of opportunities for microbiology. It's a lot of opportunities. I'm sure even in India, the microbiology is one of, now you can see like the COVID-19. So there is a lot of research scopes. I'm sure that will continue even after the vaccine comes. So microbiology has a lot of potential. So anything that if you need any help after MSc, please contact me. I would be happy to help. Do we get scholarship if we get through GRE? It depends on the university. Most of the time, like once you are here, like right state for every students, when they come for the first semester, they probably give like a little scholarship that may not be enough, but they will give some. Some universities, they give you the full scholarship. It depends upon your score. If you have a good TOEFL GRE score, there is a good chance that you will get a scholarship. Gene therapy can be used in discovery of vaccines. Well, these are two different things. Now, gene therapy, well, if you look at like the recent vaccine, like there is a Moderna company that they are taking the vaccine by developing the transcription like a messenger RNA. So that's actually not the antibodies based the vaccine. It is really the gene based vaccine. So yeah, there is a good chance. Biotechnology is a good area. That's a medical microbiology. It's a very good. There's a lot of scope there. In fact, in the NIH there is a special department that they do a lot of medical microbiology. The genetics, yes, there is a lot of scopes. After BSc, what is the career step in the MSc? It really depends on your interest. You can honestly, I would not think that way. MSc you have to do because only biotechnology, microbiology. I would not recommend you to look from that context. I would recommend you to look, are you really interested? Even if you do like any subject in MSc with your interest, that is more valuable than doing the MSc because of like the whole perception, like hey, this subject has more jobs, or you heard from somebody, I don't know the reality. And a lot of people, they take that kind of recommendations. I would not recommend. Do MSc with your own passion and interest. The top universities for MSc, it depends where you want to go. There are top universities called Ivy Leagues, like Harvard, UPenn, Stanford, Columbia. These are like these are one of the top universities. But then you go to the second universities. To get into those top universities, like very tough sometime. But you have to get really a good GRE and TOEFL. Those universities, they don't only look your score and grades, but they also look at your passion, your personality, your character. When you go to the top universities, they look at your holistic approach as a person, not just a career. Somebody might be too smart in the subject, but he may not be the team player. He may not have that other extracurriculum activities. So those big universities, they look at those. For microbiology students, as I mentioned, yes, there are a lot of opportunities. The internship opportunities I would recommend to try in India while you are a student. That is the best. I know some people in IICT or CCMB or in NIPER. I might connect some people. If you guys are interested, I can at least ask them, hey, these are the students, they want to volunteer. Can you allow them like 2 weeks, 3 weeks? That's really the best that you will get, like hands-on experience in a real research in the lab. How come we come to know? Well, the internship, they don't put it on the website. Usually when you apply, when you get the admission, then you have the option to apply for the internship once you get the admission. So that is, sometime like if not everybody will get the internship, the most of the people, they become more like, once you come here, you spend the first semester, the professors know you, they know how you work, how they know your grades. At that point you get more kind of opportunities for internship, but sometimes getting directly the internship is very hard because they don't know you that well. The food and nutrition, in what way it has a great future to students? Well, I mean, you probably know the nutrition has, like right now, I mean, even in India, the nutrition is really taking a lot of interest. I'm sure there is a lot of potential for you. Can we publish articles regarding the research topics, like in which year can we start to write them? Well, it's hard to publish papers while you are an undergraduate, like in BSc, but you have to do like a real research in the lab. When you do the lab, you have to get the good results, reproducible results. Getting a first paper is little, you have to have a lot of time in research. After, can we do a job in research lab after Bsc Microbiology? Yes, of course. I mean, you have, it all depends. Like after Bsc, I mean, there's a scope, but you need to realize that when you come out of the college, how much experience you have to work in the research environment. That depends. That's why sometime if you do the internship or if you do some training, that might be helpful. How can we get into the research field while you are a master and these are a BSc student? That perhaps, you can talk to your professor, lecturers, or the whoever teaching, they might be guide you, but mostly I believe that you have to contact the universities where there is a active research lab. Go talk to them. I'm pretty sure that they allow to volunteer. I mean, they always look for some volunteering students. So that is the best thing to start with. After microbiology, for MSc, there are a lot of, I mean, you can continue MSc in microbiology or you can change your subject. So university regarding MSc after any university is probably as good as in India, I mean, I don't think that if you work hard, if you are really interested in subject, you can pick up in any university. Because right now there are so many universities for master, because back in 90s when we were there are only like 2-3 university in the whole AP. That's a good question. So how did I adapt it to focus towards pharmacology, toxicology, nanotechnology, and honestly, right now I am more work I'm doing on like I just got a project on the quantum biology, so it's more related to quantum physics. It just, you as you go with the experience, You read the papers, you learn, you get the ideas. What is the question? The question is regarding life sciences. But in order to in order to get the right answer to that question, you may have to use different tools. You may use the tools in physics, chemistry, or you may have to use in chemical engineering. So there are so many tools that you can use. And in fact, there are two of my PhD students, they are chemical engineer. One is chemical engineer, another is biomedical engineer, because I want the biomedical chemical engineers to devise some, to build some devices for the exposure. So because they're engineers, they're the right people. So, I mean, they're working in a biology now. So it's a really the combination. It's not the degree what you have. It's the what you want to do that. What's the question you have in mind? How you address that question. The subjects is like a tool. It's like a toolbox. We have to face many failures, and many of them don't believe us. Like we face many. Yes, sir. That's a very good question that someone asked. The particularly theme, we have to face many failures. Absolutely. Each time you fail, do not take that as a negative, because every time you fail, you get a lesson. I mean, that's the key thing. So failure is not something bad. Don't take that like, hey, I couldn't succeed this year, but really encourage yourself and take a positive step back and think positively, and do not stick to that failure. That's not the right attitude, and always look forward. I mean, I know people discourage, ignore them, and try to focus and bring your unique thing, whatever you have. Definitely you have a uniqueness in you. You need to recognize that. What the scope of narcotics? I don't know if I understand that question correctly. I mean, are you talking about the research, or it is something? Is it a long time to settle in research field? Well, I mean, it takes the time in any field. I mean, let us take you, you're not in research, you are in the administration. You're administrative officer. Don't you think it takes time to settle down in that? You're not going to jump at the higher level right away. It takes time. You need to learn. You need to do good job. You need to get the appreciation from the society, from the customer. Then only you go in any field. So that is not only in research, in any field.
I think no other questions.
Okay.
I request Mohammed Nazneen to propose formal vote of thanks.
Ma'am, am I audible? Hello?
Yes, ma'am. You're audible. You're audible.
Okay. Respected and most distinguished speaker, Dr. Saber Hussain, it is really an honor for us to have you with us this morning and probably an evening there. It seems to be a great honor to propose the vote of thanks to all who have helped us. I'm so sorry, sir. There are elections going on here. People are very excited about them. It seems to be a great honor to propose the vote of thanks to all who have helped us in making this program a success. Firstly, I'd like to give my hearty vote of thanks on behalf of our entire college to Professor Saber Hussain for gracing today's program. Sir, your words today, we are very confident that they are going to pave the way for our students in the future. And we definitely expect them to come up with flying colors. Thank you, sir, for your interesting, thought-provoking address. It is my pleasure to thank our principal, Dr. Achuta ma'am, HOD, Dr. Pradyuta ma'am, and our all the faculty members who have contributed and helped us to make this program a success. Thank you, ma'am.
Thank you, sir. Thank you very much. It's too late night, so we can close the session now.
Thank you. I appreciate.
Girls, hope this webinar is definitely very helpful to you. So now you can go to attend the classes.
Ma'am, shall we end the session, ma'am?
Yeah, Shruti, I'm ending it. Yeah,
Okay.
I'm stopping live streaming also.